thank you. Thank you very much for this kind invitation to give this presentation here. So today, this joint presentation with Hongbo and me will work like this, that I will give a quite brief and short general introduction to our activities that do relate to nanomedicine. And then Hongbo will move on to a slightly longer presentation showing you all this fancy stuff. So please do not log off, even though you find my part very, general or easy or boring or such. I promise you that Hongbo will show you all the fancy stuff later. And just because I had the opportunity, I couldn't help myself but seize this moment to have a short commercial break that I thought was very relevant for CRS Nordic and Nordic POP, which is that we have now been educating pharmacists on bachelor level since 1956. Here you see a very nice magazine article from 1959 when 5 of our first pharmacists graduated. And since we do this fully in Swedish, we take pride in that we kind of open the doors to the Nordic countries. And this is also currently in the strategy of our university, that we want to be the Nordic gateway to Finland, or Finland's gateway to the Nordic countries. That way it would be. And of course, being in Finland, actually up to 1/3 of our undergrad students are also Finnish speaking. There are quite stringent language requirements to get into study, but so far, I mean, it's working. I know of very many of our undergrad students that have gone on to find work in especially Sweden and Norway. So end the commercial break. So today we were supposed to be talking about research, and these are our research groups and what we are doing. This is not the most recent slide, but I still wanted to include it because I'm sure many of you know Niklas Sandler, who was with us until up until a couple of years ago when he chose to join the dark side. So he moved fully to industry, and his main company is also very relevant to today's topic. So he works at Nanoform. He also has other spin-off companies like Purify and so on. And we still have some of his activities also on printing going on at our unit. But if you go to our website, this would be the most current setup of PIs and research groups. So the newest PI here would be Tappi, who joined us from September last year, and he has a shared professorship together with University of Helsinki and Åbo Akademi University on novel drug formulations within the national FinPharma network. And out of these groups, it would maybe be us three then that are mostly working on activities relevant to today's topic, which is nanomedicine. We have also a couple of joint projects which are you nowadays, so we are also luring her into this wonderful world of nanomedicine. So this would be a slide where we have tried to kind of combine all of our activities into one. So we are in general working on functional materials and technologies for drug delivery, but also biomedical engineering. So what many of us are doing is developing novel drug delivery systems for all kinds of different active agents, from small molecule agents to biomolecules. These would be based on new carrier materials that we either develop ourselves or together with collaborators, and maybe different kinds of polymers. They may be inorganic materials, hydrogels, which are mostly polymers with a lot of water, and of course, nanoparticulate systems. So we're also looking then into new technologies in drug manufacturing, again, those being different printing technologies, of course, nanotechnologies, for instance, microfluidics for manufacturing different nanoparticles, electrospinning, and so forth, of course, depending on what kind of dosage form you may be designing. And of course, we want to understand what's going on and how our systems are performing. So we also have biological lab spaces where we can we can perform at least the most basic cellular and bacterial works, but if it gets too complicated, we have cell biology on the floor right beneath us, or of course we may do this with other collaborators. For instance, Hongbo works actively with China, that I'm sure he will show you soon. And also Outi, who works mostly with computer-aided drug design. We have also lured into this wonderful world of nanomedicine, in that we have recently starting to collaborate on computational pharmaceutics, including nanoparticles, and it seems to be explaining a lot of things. So we are very happy with this recent development also into the world of computational methods for explaining what's going on in nanomedicine. Then we have a few people then working more on the biological side of life. We have, for instance, Tuomas Näreoja, who is developing different kinds of 3D disease models. Xiao Yu, who works mostly with bioprinting, also for making different types of models or tissue engineering. And microfluidics can not only be used for manufacturing different type of nanoparticles, but it's also good, for instance, for single cell RNA sequencing, single cell analysis and screening, droplet-based assays, and on-chip screening and diagnostics. Again, I'm sure Hongbo will show you some fancy examples of this. But if we're sticking to today's topic, so I myself have been working in the wonderful world of mesoporous silica for 20 plus years now. I think I graduated exactly 20 years ago with a master thesis that dealt with mesoporous silica for drug delivery, and at that point in time they were still not in nanoparticulate format. That came about 10 years, or not, but almost 10 years later. And I'm still sticking to this within our research group because it's a very useful platform to be working with. It's suitable for virtually any type of active agent delivery within the small molecular agents. Especially hydrophobic molecules are very feasibly formulated with mesoporous silica, and they are also very suitable for delivery of biomolecules, being ceramic matrices, so they provide very effective and good shelter for these fragile molecules. And being porous particles, so you pack the active agents into the pores, you can also, at least in theory, also kind of making it in reality, design quite fancy, sustained, or mostly controlled release mechanisms for instance, having these molecular gates that release the active agents out from the pores at will, and so forth. So they have proved to be so useful that I'm still 20 years later sticking to those. And of course, then entering into the nanoparticle world, also with mesoporous silica. These are, of course, then subject to any kind of targeting strategy that any other nanoparticle system would also be. And since a nanoparticle or nanoparticles as such may not be in that sense a final formulation, we are also looking into how to make these into viable products. And this, of course, the final dosage form here would, of course, then very largely depend on which route of administration would be applicable for this particular nanomedical formulation. IV is, of course, a given. That's still the most common one for administering nanomedicines, but we have also looked into oral, topical, and pulmonary formulations, and even nanoparticles for crossing the BBB. And dosage forms that we have made to date include then dermal patches, antibacterial coatings, orodispersible films, and different types of printed formulations. And since I know this audience here may be quite interested in especially this field, I also included a couple of examples here. So, for instance, if you are dealing with a challenging molecule that you would want to print, challenging in terms of, for instance, pore solubility or very high potency, it turns out that nanoparticles have the perfect size for instance, inkjet printing. So then you can instead load your challenging molecule into these nanoparticles and formulate your nanoparticle suspension into an ink instead. And again, then in the best case, have a very functional ink where you even then built-in, sustained, or even controlled release into these particles. Your ink will possess these properties. Similar approaches can be included also in tissue engineering, because we know how to design the nanoparticles so cells will like them and take them up. So this way we can, even in printed scaffolds, even if we are doing cell-laden bioprinting, so printing cells into the same bio ink, we can also mix in then loaded nanoparticles, and in the final scaffold then have these cells hopefully then proliferating and developing in the right way that they should if dealing with tissue engineering. Yes, and since Tapp is not presenting today, but he's still doing relevant activities, I also included one slide of his activities. So he is a physical chemist just as myself. So he likes surfaces and interfaces, and that's also what he's doing in his research. And he has these fancy technologies, for instance, SPR or surface plasmon resonance, I think it stands for, that enables him to sorry, lunch clock here. Yes, so in real time following, for instance, drug and nanoparticle action and efficacy together with cells, or they can also be different types of, for instance, printed structures, may it then be carrier matrices or scaffolds or in vivo models that these interactions are being studied with. And a current recent interest that I know he's actively looking for financing for is to try to determine plasma protein interactions with nanoparticles in greater detail that perhaps anybody has done before. Again, since his technologies and methods enable him to really in real time monitor plasma protein interaction and absorption with nanoparticles or any other types of surfaces. And the same methods can also be used then for more biological studies. So also in real-time monitoring cell and bacterial interaction, absorption, proliferation, or even bacterial biofilm growth to see, for instance, how a biofilm would react if you introduce it to different types of drugs or nanoparticles or drug-loaded nanoparticles. And yes, as general point, of course, Turku is a nice area to be doing this. About half of Finland's pharma and diagnostic industry is located here in the Turku area, with the, for instance, Bayer that is here is actually Bayer's, I think, third biggest plant, and they recently also invested 250 million euros to expand it even more. And about 20% of the industrial workplaces and turnover in this area come from biotech companies. And after a couple of, or even a few years struggle, it was also now, or planning of this initiative, it was recently also established that the National Drug Development Center will be placed here. And I think we will have some kind of headquarters here in BioCity, where we also are located. So due to this favorable environment we are working in, we also have a joint the Akademi of Finland financed flagship together with our neighbor University of Turku, focusing on the immune system, and Hongbo is a affiliated PI in this flagship. So again, I'm sure he will also show you some examples that will relate to this. And that concludes my introduction. So you can find us on the platforms you would be expected to find us. And if you don't have any very urgent questions on this general part, I invite Hongbo to now take over. So thank you on my behalf.
Happy to give a talk in this network. So we haven't been very active in this Nano-Pop event due to this panic situation, but hopefully this will be better and we can be active again. But it's also very interesting that we have many webinars and we try to collaborate with each other and also promote the communication with each other through internet. So today I will tell something about a solution for health with functional material. let me see whether yeah, now I have the laser pointer. Yes, so I will first tell about how we develop this material, and we will give some examples on how we utilize them, and our aim is to have some clinical translation. So that's why we also have close collaboration with many hospitals in China and also some hospitals in Finland and other countries. So we work on different, we use different types of technology, including microfluidics, bioprinting, electrospinning, and we also make bulk fabrication. So with all these technologies, we can produce material from different scales, starting from the very tiny ones in nanoscale, and also the big ones in even centimeters. So as you can see, here are the nanoparticles produced, and we are not a true material group, but we utilize different kind of group, different kind of materials as our toolbox to treat some different diseases. And here I want to give some examples on how we utilize different kind of material. So the first type of material is the DNA-based nanomaterials. So for this type of material, you can produce a different kind of shape by designing the DNA strand in a certain way. For example, here we can make this kind of shape. And then since the DNA is also, there is some functional group you can use to further modify them. And there is also possible to conjugate some detection ligand on top of this structure. And then you can let this structure sit on a surface. So why we do this kind of design is because for diagnostic, if you directly throw this probe into a solution, then this probe will be everywhere. So it will, they may also aggregate with each other. They may attach to the bottom or to the surface of the container. In that case, you lose the detection activity. But if we have such kind of structure and then we fix this probe in this direction, in that case, the probe is always free. And we can have 100% efficiency. As you can see in this example, we have used this kind of structure to fix probe in a tube. So, and we can use different probe to detect different biomarkers like ATP, cocaine, therapy. And they are the examples. And then when you flow a liquid through this channel, you will have signals from different region. And we also do some kinetic analysis to see this detection. And also we have do the selectivity analysis and binding kinetics. So the results shows clearly that our method is very sensitive for detecting different biomarkers, and also the selectivity is very good. And there are also some other ways for making precise detection. So in this case, we can build up a kind of molecular imprinting member. So for this member, it's like if you have a compound of interest, then you will try to make use different modes, and then there are different chemicals. Then you can fix this target molecule into a kind of cavity. So in this way, you are building kind of detection member, which is very selective for this molecule of interest, because you design this member based on this molecule. And also you can also add some fluorescent probe, for example, this one, into this structure. And in that case, when you have a detection molecule which dock to this pocket, then they will have a molecule, this fluorescent quenching effect with this fluorescent probe you put in this structure. So in that case, the fluorescent signal will decrease. And by calculating the fluorescent change, you can quantify the amount of molecule detected. And another important thing is like this member is very thin normally, so they are not very flexible when you use them. So in that case, we often use silicon oxide particle as a core, and then we also, in many cases, use silver or some other nanoparticles to have a better electronic condensing. So we use this as a host for this member to be stable. So as you can see, here is this structure we produced. And then here again, we show this detection sensitivity and selectivity. And we use the swan urine, this biological sample, to have this kind of testing. And for nanoparticles, we can also use them for precision labeling. For example, in this case, we are thinking to label the tumor. There are many these medical imaging tools that you can observe a tumor in patient, but those tools are normally a very big instrument. And in that case, it's difficult to use them in the operation room. So when the clinical doctors are doing surgery for this tumor, nowadays people are still using naked eye to try to identify where is the tumor. So that's why we are thinking if we can develop a method that we can label the tumor, and we can use a hand device which can be carried inside this operation room to detect the signal, then this will be very useful for clinical application. So in this case, we have been using this gold nanostar particles, and we also use probe, DTTC probe, and this will have a Raman signal. And for Raman signal, the good thing is, as I said, you can have a very small hand device to detect Raman signal. So in this case, this is applicable in operation room. As you can see, when we inject these nanoparticles through the tail of the mice, they will accumulate in the tumor site, but they don't accumulate in the skin beside this tumor. The reason is because for cancer, there is a theory called EPR effect. If the nanoparticle is around 100 nanometer or a bit smaller, so they will have a better accumulation in the tumor site because the tumor vascular are more leakage, but they will not accumulate in the skin because the blood vesicle is more close, so they will not pass through. So as you can see here, we can clearly distinguish the boundary between the tumor and skin with such kind of nanoparticle and Raman labeling. And then the clinician can do operation based on this signal. So we also do this original tumor at the original site, this colonic tumor, and we do this operation in live mice, and then we close the wound, and after 2 months, we can see that the tumor is not coming back. And we can also do stem cell labeling. For stem cell therapy, it's also a hot topic. But the problem is like for stem cell, it's difficult to estimate what happened after we inject them into the body. So it's very important to track how they go and also to regulate the differentiation after they are transplanted into the patient. So in this case, we can produce a nanoparticle, and here this nanoparticle has a photoacoustic imaging property, which is also a commonly used instrument you can find in hospital. So in vitro, we can see that when we incubate this nanoparticle with the stem cell, they are uptaken by the cells. And by controlling this concentration, so we can find a very good concentration, which have a good labeling property, but has no harm to the stem cell. And also we can load, because this is porous material, we can also load some important markers into these pores, and then this can regulate this stem cell differentiation. So as you can see, now we have loaded this WNT3 alpha into this porous particle, and this protein can prevent this stem cell from oxygen stress. And we also do different kind of biological analysis. So from this, you can see that we have injected this nanoparticle into this the heart of the mice, and as you can see, even after 28 days, we can still track this signal. And here we can also use nanoparticles for cancer treatment. So in this case, we have applied a combination of a metal-based particle with a biological-based member. So we first incubate the porous silicon nanoparticles, which is loaded with a cancer drug, with the cancer cell in vitro, and then we will break down this cancer cell, and then we isolate back the nanoparticle. And as we can see, after this isolation, we notice that this porous nanoparticle is coated with a member. And then we start to analyze all these biological processes. So we first analyze how those particles are uptaken by cell, and we clearly see they are following this autophagy, this pathway. And also we need to investigate what indeed this layer, this coating layer it is. So we do different biomarker analysis, and then from this biomarker, we can identify that this member is more like an exosome marker. So we also check the drug loading and also this drug release. And in vitro, we study this cancer cell inhibition. And then we also do this in vivo study to see how they behave in the tumor treatment. So as you can see that when we have this exosome coating, we notice that this particle ultimately accumulate in the tumor, and also they have a deep penetration into a solid tumor. And this is like, we don't know the mechanism, but this is very interesting finding. And we think this is very important for tumor treatment, because in solid tumor, there is a stem cell theory. And the cancer stem cell are very important for tumor to grow and to migrate. But the cancer stem cell is normally very deep in a solid tumor. And for normal drug delivery, it's not easy to reach that region. But with this, they ultimately reach very deep in a solid tumor. And from this tumor inhibition study in vivo, we also noticed that even we use much less of drug, we can have a very good tumor inhibition effect with this nanosystem. So, and we can also do another type of exosome-based design. So in this case, we use neutrophil cell. So we first isolate this neutrophil-derived exosome, and then we also use another way. So we extrude the whole neutrophil cell to a small pieces, and they will self-assemble into the vesicles. And then we notice that such kind of vesicle has a exosome-like property. So as you can see from here, so we produce both these, we both isolate this natural exosome, but also we produce exosome mimetic structure vesicle. And also to better regulate this target delivery, so we can also have a supermagnetic particle functionalized on top of such kind of exosome. In that case, you can drive them better into the tumor site. And here, the advantage of using this modified mimetic exosome is because you will have much better yield by this method, and also you can load some drug, for example, this doxorubicin, into this kind of self-assembled system. So as you can see, here is the material production. So we can produce both, we can isolate both these natural exosome, but also we can produce this mimetic vesicle. And we also do this in vivo study to see the behavior and also this tumor inhibition. So we can see that with this natural exosome, they have somehow tumor inhibition effect. And then if you conjugate this supermagnetic particle to regulate them better to this tumor site, then you can have a better inhibition. And interestingly, if you use this doxorubicin loaded mimetic vesicle, you can have even better tumor inhibition effect. So we also study this in another cell model, this mice model, NCC mice, and we have the similar finding as the first mice model. And we can also use some other way to regulate and to improve this tumor penetration, because as I mentioned, to penetrate deep inside the tumor is very important for effective tumor inhibition. So as I mentioned, to accumulate the nanoparticle in tumor, you would need a nanoparticle which is 100 nanometer size. But if the nanoparticle is very small, for example, if it's less than 10 nanometer, then they are too small that they can be easily eliminated by kidney from the body. So in that case, it's also not good. But in order to have a tumor penetration, then the 100 nanometer is too big. So they cannot penetrate deep inside a solid tumor. So in that case, you will need a smaller size. So that's why we have this kind of design that we can have an in vivo size transformation at the tumor site. For example, as you can see, here is the nanoparticle produced. So we have some photothermal responsive particle, and then we load them into a polymer matrix. And then this will become 50 to 100 nanometer size particle. And you can also have some drug into this molecular, this polymer matrix. So when they reach this tumor site, you can apply a laser, and then this photothermal responsive particle will generate some heat, and then this polymer matrix will degrade. In that case, you will release the smaller particle. And then they are carrying heat, so they are very molecularly active, and they will better drive this drug, doxorubicin, to go deeper inside the tumor. So as you can see, by this method, we can clearly see that we can inhibit the tumor in efficient way. So, and in many cases, we also need a combination of different drugs. So it's not easy to co-load many drugs together, because they have different pharmaceutical properties. For example, some drugs are hydrophilic and some drugs are hydrophobic. So it's difficult for them to be co-delivered. So in this case, we are thinking, is there a method we can, which is broadly applicable for loading all kinds of drugs? So here we have been using microfluidic technology. So we have produced microfluidic-based double emulsion. And in this double emulsion, you can have a water core with kind of hydrophobic shell. It's two layers of droplet. And then for this kind of structure, you can load hydrophilic stuff inside and hydrophobic stuff in the outer droplet. And we make such kind of droplet using hybrid polymersome and liposome. And the good thing to use such kind of structure is because they have a self-assemble property. So when we have this very big droplet, which is in micrometer size, we can extrude them through a membrane, and then they will become very small nanoparticles or nano droplet, which is injectable to the mice. So as you can see, first we produce this double emulsion. There's 2 layers of droplet, and then we extrude them through this membrane, and they will become self-assemble into this nanometer size droplet. And with this method, we can load all kinds of drugs, because you can load both hydrophilic and hydrophobic stuff. And we noticed that if we enable the combination of a series of drugs, you can have a very good tumor inhibition. And this is another example on tumor treatment. So instead of tail injection by multi-doses, it's also possible to have 1 dose of treatment with a local minimally invasive injection. So in this case, we have first have this gold nanorod, which have photothermal responsive, and then we grow MSN shell on top of this gold nanorod. So in this case, this shell is porous. It can load different kind of drugs. And we can also modify the surface freely, because this silica oxide is very easy to be modified. So you can have some ligand. And then when you have this nanoparticle, you can see them into a micrometer hydrogel. And you can produce such kind of hydrogel through microfluidics. So as you can see, here is the material produced. So this is like a gold nanorod, and this is with the this MSN shell. And we test this photothermal responsive. And then with microfluidics, we can produce these microparticles at different size. And we choose the proper size for the next application. So as you can see, this is like the droplet we make, and then we load these nanoparticles inside this droplet. So as you can see, these nanoparticles, they are the very small, tiny green dot. So they have an equal distribution in this matrix. And for this release, we can have a really sustained release because of this hydrogel matrix. In 24 days, we can still have some drug remaining. And you can promote the drug release by apply laser at different time point as you wish. For example, here we apply a laser at 6 day and 12 day, and at each laser point, you are stimulating this drug release. And then we have inject this hydrogel, microgel-based particles locally into the tumor. So with one dose, they can sustain release the drug for a very long time. And interestingly, we found that we can clearly reduce the systemic side effect of drug dose. And also, even with 25 times less of total drug dosed comparing to this tail injection, we can have a better tumor inhibition effect. So this method is very effective. And we also do some tissue engineering-based project. For example, here we use 3D biomimetic material for bone regeneration. So for the natural bone, they have a unique structure. And we are thinking if we can highly mimetic this structure, can we promote the bone regeneration without using any drug? So here we first make this seal fiber as a scaffold. And this scaffold is mimicking the hard part of the bone. And then we seeded this bioglass into the structure, and they are mimicking the nanoparticles in the bone. And then we put this collagen to fill this structure, and collagen is mimicking this a bit softer part of the bone. So as you can see, these pictures, we can see like this is a cell fiber, and the one seeded with this bioglass, and then with collagen network. So after we build up such kind of structure, we culture cell on top of this scaffold. And clearly we notice that the better you mimetic this structure, the better it can promote the bone stem cell to grow. And we test gene expression to see whether there are important genes expressed. And again, we find the similar finding that the better you mimetic the structure, the better it can promote this positive gene expression. And then we also check the mechanical property. So we notice like if you seed this collagen into the structure, it can significantly improve this stress stand for the bone, for this structure, which can better mimetic the bone. And we also use animal models, and we can see that when we highly mimetic the bone structure without using any drug, we can already promote the bone regeneration in a very good way. And the material we used are biodegradable. So when the new bone grows, this material will degrade. And also we can use DNA ink to produce different kind of wearable device. So in this case, using DNA chemistry, we can build up a spatial structure. For example, as you can see from here, we can make such kind of structure And then we can insert some enzyme like hemine into this structure, and they will grow up in this way. So as you can see, this is like the fiber-like structure we produced using this DNA chemistry. And they have a polymeric and kind of property. So if you put them in water, they will become hydrogel. And for the enzyme used, they can have they can catalyze some oxidation reaction. So in this reaction, they will generate some oxygen. But for oxygen, you cannot directly detect them. You need to convert them to another kind of detectable signal. So in that case, we have coated this with another layer of polymer, and this polymer can convert this oxygen signal into a hydrogen ion. So in that case, the hydrogen ion can be detected as an electronic signal. So because this is like a hydrogel structure, so they can be used as a flexible ink for 3D printing. So you can print them into a different kind of structure as you wish. And then you can see some like GOX, this GOX enzyme into this structure. And then with this hemine and GOX, when there is glucose detected, the reaction will happen, a serial reaction will happen, and in the end, you will have a electronic signal. So in this case, you can make a flexible wearable device for glucose detection. And recently, we have also been writing many review papers because there are some period we cannot do very much experiment. So if you are interested, you can read some of these review articles we wrote. Okay, thank you for your attention.